you can take the in vivo. So in vitro followed by in vivo, then followed by you prove the efficacy and the safety, then it will be going to the clinical trial. That's how the plan will be, right? Then based on whether it's a successful or not, it will move into the next stage. And it's that a drug can be failing at any stage. That's how it is happening, right? Next slide. So this is another way of showing it, right? You have a pre-discovery where you do the basic research. In a sense, when are you talking about the drug discovery? How do you start it? You suddenly to pick a molecule and do it? No, right? There has to be a lot of background work needs to be done. First of all, you should decide what disease you want to talk about. There are so many combinations, right? Do you have the drugs for that? You may have the drugs. There is a need for to discover another drug. What is the unmet need? You maybe have an unmet need, but do you have the tool to do it? You need to have a model also, right? You need to have a proper screening tool. Then only you will be able to say that, no, I can do the discovery. So a lot of things needs to be kept in mind. You need to have a proper assay tool. Maybe coming and giving you that I could not take the molecule because I could not prove it experimentally because the tools were not supporting me. And the other products, the natural products are mostly polypharmacology. They do multiple pleiotropic effects. That was so reason. So that means it has to be very targeted discovery. That means you know the target, you know the disease first, then you know the target, and you know the reason why you are taking the target. The reason for you to take the target was it is very selectively expressing the disease. That's a good target, right? It should be only expressing the disease. Are there any targets like that? I don't think so. There may be related expressions altered. Normally it may be this much. In the disease, maybe that's a good target for you, right? And that should not be having a very significant physiological effect. What will happen? What happened to the roficoxib? Celecoxib, roficoxib. What happened to that? Do you have that drug now? Roficoxib. It's not there because it's been withdrawn because it is COX2 inhibitor. We know COX2 is the selective inhibitors are very good, but the same COX2 inhibitor is also causing cardiotoxicity because the COX2 enzyme was required for you to see the cardiac function. So it's actually causing abnormality. So that means the target which you thought is a very good later become not a very good target. So use biologics, maps. It's the era of the maps now. What are the maps? Monoclonal antibodies. Can we dream of doing it? What are the things required? How many of you know what is the hybridoma technology, right? You might have studied in B pharmacy. Hybridoma technology you studied with the difficulty. Okay, this is an important question because you're going to be having in the exam. The same technology, if you have a good basics, you can have your own company. Trust me. Of course, you'll be. No, if you can produce one monoclonal antibody, new monoclonal antibody, which is going to target your receptor, you don't need 8,000 crores of money. Just need initial studies proving that, okay, I have this monoclonal antibody, effective in my animal model, collaborate with the biotechnology people and say that, no, this is my initial proof of concept. Now, whoever want to collaborate with me, people can come to you. What is the good thing with the biologics? They are going to be very selective, specific. They don't produce a lot of toxic effects. There are issues with the manufacturing characterization, but otherwise, these are very selective. They produce a very specific function. That's how the monoclonal antibodies can be one of the function for us. But at least, do you know the procedure? How we produce it? No, we just studied and forgot, including we all the faculty. So these are the things really will help you tomorrow. Go to industry, work for some time, have enough basics, build your own business plan, come out and start it, right? Whoever is going to work tomorrow for 20 years in the same organization is a failure career tomorrow. You need to have your own startup, and this startup should be originated for your basics. That's what I'm trying to make it here. So phase 1, phase 2, phase 3, I don't want to talk about it. You all know it, what exactly you do it. So let's move to the next stage. Even drugs can be withdrawn from the market after approval, right? Can you give the example? I just gave an example of roficoxib. Can you give other examples? Drugs can be withdrawn from the market even after approval. Example? Thalidomide. Yeah, exactly. So that was a 1950-60 story. Even I have a drugs like terfenadine, which was an antihistamine drug. If we been withdrawn because of the QT prolongation, also called torsade de pointes. The classic cisapride. These are the drugs withdrawn from the market because of the safety issues. They always say there is a risk versus benefit. That means even drug is not even safer even after approval. You need to keep on producing the pharmacovigilance data saying that no, Always risk is lesser compared to the benefit. Then only drug will be surviving, right? So nothing is actually certain till you prove the data every year, right? Next slide. Next slide. Okay, thanks. Sorry. I mentioned target validation. I may be taking little faster time, a little more time, but don't worry. You have full day, right? You have full day to do your all the activities. I will also visit your stalls, right? It's just first hour only, right? 10 o'clock is the first hour. You should have enough time, right? So I may be taking more time, so I'm just making you mentally prepared for that, right? Yeah. Objective interacting with you is maybe some of you may be thinking differently than what conventionally you have been thinking of. And some of you will be definitely getting some hints from what discussing today, right? So these are the step 1, 2, 3, 4, whatever it is. Target validation, very important. How do you validate the target? And see, you have 10,000, 20,000 molecules. Lot of compounds, right? You have how many of you with the chemistry background? Some of you, right? One synthetic route, how many times? We may be having multiple steps. You need to have 10,000 molecules synthesized. How much time is required for you to even get the drugs? NCs. It's a lot of time, right? That means that's why it's risky. 10,000 to 20,000 molecules. A lot of molecules needs to be having it for you to have one successful drug. So that's one thing. Now that means if you have to synthesize this many number of molecules, what is a good target for you? Do you want to take the G-protein coupled receptor? Let's say G-protein coupled receptor. How did you discover the alpha 1 receptor antagonist or beta agonist, beta agonist? How did you discover it? Did we ever imagine how did they discover this? How do you know that these particular compounds are binding? At that time we have been doing this. What is that? Isolated tissue experiments. Isolated tissue experiments, very classical pharmaceutical experiments where you take the tissue and mount in the organ bath and add the agonist, antagonist, will come to know. There was a Nobel Prize also given in that particular same kind of discovery. Robert Fudge got, who is a pharmacologist, for nitric oxide discovery, for endothelial relaxation, he got the Nobel Prize. Based on that, we have nitroprusside and all the drugs have come for the angina and other treatments, right? Yeah, with the conventional pharmacology, people have discovered, but that was the era at that time. But today we in a different era, right? Now you need to have modern tools. How do you able to prove that your compound is binding to your receptor? That means your GPCR, what are the GPCR you are talking about? You should have the crystal structure. You should know which exactly the binding site. You are saying 7 transmembrane domains. Have you seen it? Textbook you have seen it, but how do you explain the prove it? So not so simple, right? One protein molecule, that too having a different membranes and also having a binding site. So these are a lot of modern tools people have to use it. These all the structures can be proven through the AML nowadays, right? Now, once you have a target validated, then you will be trying to understand whether this particular target is having a role in the disease or not. And again, you can use some of the informatics tools as well as any other omics tools to mine the data so that okay, this is the target found to be having a disease. How do you do it? You have a disease tissue, you have a normal tissue, you compare it. There is a pattern happening. Okay, in that particular pattern, there are group of proteins are altered, group of receptors are altered. Okay, can I take this particular receptor? Can I take this particular target? Now you try to validate. How do you validate it? You try to knock down the particular target. Or you try to overexpress the target by using biotechnology tools, so that your disease should be decreased or increased based on what you are doing it. That means, okay, this is a proper target. Now, again, go back to human samples. Test in the tissues what happening in this particular tumor. Check the tissues what happened to this particular protein. You can do the immunohistochemistry and other characterization techniques, so that you will exactly understand, okay, not only in the animal, in the clinical samples also this particular protein is having a role. That means it's a good target for me to explore further. And simultaneously, okay, what is its role in the physiology? If it's alter the physiology, it's a big problem. We don't want any target to be even also having a physiological role. If it's having a physiological role, it's gone. Your drug will be showing a later problems during clinical trial. That's a big costly failure for us, right? Lead optimization. Once you have a target, you try to optimize the lead. What is exactly? If there is a hit, there is a lead. Hit means it's just going and hitting it, that's all. But it may not be a optimal molecule. Now you need to make a lead molecules from that. That means I try to modify the structure. That's what the SAR, structural active relationship. You try to modify the chemistry. You try to modify even the heterocyclic if required. Then you try to come up with a a molecule, probably a lead. How do you know it's a lead? Then we'll be doing the in vitro studies or enzymatic assays, saying that okay, it's having a proper interaction with your target. Now that means it may be probably a active compound. Then we will be trying to do the in vitro activity studies followed by in vivo activity studies. Along with that, safety data also generated. Safety is very, very important, right? Without safety data, none of the drug will be going to the next level. You cannot even think of even going for IND filing without a safety data, right? Safety is utmost importance. And if you see the ICS guidelines, QSEM, quality, safety, efficacy, multidisciplinary. Safety guidelines are very, very important for the INDs, right? So see the number from 10,000 molecules to 250 molecules to 2.5, 2 to 5 molecules, and only one drug may be successful, and there is a very good chance that drug may be failing at the phase 3 trial. If the drug is failing at the phase 3 trial, it's a multi-centric. You might have spent already 10 years, all your $2 billion are gone. Only major pharma companies like Pfizer, Bayer, Eli Lilly, these are the companies can able to survive that. None of the Indian pharma company are in a position today to survive this, right? That's one of the limitation of it. These all the tools are used. I'm sure we have been hearing about this particular different terminologies at different levels. Maybe in B farm level, maybe some of the M farm level. The all might have studied in silico. We are talking about cellular genetic targets, genomics, proteomics, bioinformatics. Nikhil was working in this area. So every field is important. It's highly multidisciplinary in nature. You cannot just a pharmacologist cannot bring a drug until unless he has a right team of people working in the team. So it's very important to have a multidisciplinary approach. Then only you will be able to progress it. Again, as a organization, one organization cannot produce an entire drug. It's just only partly you can do it, and later you have to pass on to next team, right? So animal models are very important. Then only you will be able to prove that the compound is active or not. Sometimes you can also do in ex vivo studies, right? See the left side, it's a drug can come from biological products or the chemical synthesis. Whatever it is, what you prove is the compound biologically active? One question. Other one is the biological. Sorry, man. Okay. If the compound is safe, you see the activity, safety, then you will have the results. You'll be having results. Will be having a team meeting. Then whether to go or not go will be decided. So see the and I said the kill the compound. Killing the compound early is the best approach. I don't want the molecule to fail at a later stage. If the compound is failing after 5 years, your 4 years time is gone. In the corporate sector, it's not so simple. Everybody will be not just salary, right? So many logistics involved in that. Even electricity bill, everything is important, right? So it's a very costly affair, right? That's why they try to kill the molecules early. If you have to kill the molecule, now if I can kill the molecules by using my AML tools, the best approach. So don't do the experiments. Just put on a computer. Do automation at the computer level, saying that no, okay, this compound has this particular problem. I'm sure this is going to fail at the later stage. If you have a scientific rationale to prove it, they will actually definitely do that particular activity first. Okay, this person is raising a concern of this particular toxicity. Let me prove experimentally. Okay, if it is showing the toxicity, that means you are stopping the molecule at the early stage, not waiting for the 4 years. That's how you can actually do this. Since it's a very risky business, AML, early stage of it, it's the protein structure. Like AlphaFold is a AML driven the tool. Proteins will have so many amino acids, right? Can you remember the sequence of the proteins? But if you give the computer, it will remember, right? Which number, which amino acid is there? What are the interactions there? It's all computer can able to remember. That's what the artificial intelligence, right? So that means we can able to make a protein structure by using a artificial intelligence, not humanly. Human knowledge is required, but since human mind cannot remember everything, you are putting this data into the computer, it is going to give the protein structure. So you can understand the target properly. You can also understand the pathways. Each cell may have 10,000 proteins to 20,000 proteins. How these proteins are connected to each other, that's what informatics is all talking about. We can able to understand, okay, if I disturb this particular GPCR, if I inhibit this particular enzyme, what kind of far effects I am going to see it at the nucleus level, at the membrane level, whatever it is. So that means you will be able to understand the pathways properly, but AML tools, not just experimentally only understanding some other. That means everywhere it's going to be useful. AI deals with the simulation of human intelligence in missions programmed to think and act like humans. So you only have to train them, or you only have to give them the data sets. So it is going to make algorithms. The algorithms will become a set of results, right? It reduces the cost and reduction. Okay, now if you can reduce from 10 to 15 years to less than 5 years, 6 years, or less than even 3 years, it's a good discovery, right? A faster time. You don't need a lot of people, and also lead discovery will be much faster. Improved preclinical testing is possible, and efficient clinical trial. All the pharma companies are trying to use the clinical trial because each patient will be having a lot of huge amount of the data. Now, impossible for a humanly to handle all the data. Now you put the data into artificial intelligence tools, it is able to make a patterns. You will remember, okay, this subset of people are going to have this particular pattern of the results. So now we can able to take an efficient call whether the drug is really safe or not. We have 1,000 people. Analyzing 1,000 people data is very difficult, right? But we have a pattern how to put in the Excel sheet and put it there. It will make a pattern and tell you, okay, this compound is active or not active. Or compound is showing a particular adverse effect. Now you can understand through this particular data sets. You can do that, right? Since now see, machine intelligence, artificial intelligence, machine learning. I'm sorry, I'm also very weak in the artificial intelligence. I'm trying to still talk about this, but see, machine intelligence is something. Artificial intelligence, machine learning. Now machine is doing the intelligence. That's what machine intelligence, right? That is it. So through this particular tool, what are the failure you are seeing is the 90% failure which you may be having in the clinical trial can be reduced. It. How you are doing it? You are not applying the mission there, but when you design the molecule there itself, you are optimizing in such a way that it is trying to address all the concerns you are expecting to happening during the animal testing as well as during the clinical trial. That means, okay, is it meeting this particular safety pattern like solubility or genotoxicity, organ toxicity, reproductive toxicity? I'm trying to optimize all the things at the computer level so that my molecule will not be giving any unwanted effects later stage. That's what is the objective, right? So these are the some of the things. Many things can be done. I'm talking about the drug discovery. How many they can do? Better target identification, as I mentioned. Bioinformatics tools can be usable for you. Speed and efficiency can be increased. Hit identification, lead optimization can be much faster. Cost reduction is possible. Improved accuracy. It will be able to predict the toxic assessment and personalized medicine. I'm sure it is possible because personalized medicine, in the sense, each person will be having a certain genetic background, certain particular things. So we can actually able to profile them and only give that particular set of people with this particular drug. That's what is the personal therapy, right? These are some of the tools. The bottom one is AlphaFold. This is a Google tool, and there was a Nobel Prize given to this AlphaFold protein related discovery in the chemistry, right? Because the protein structure was so important, there was a Nobel Prize given to that. These are some of the tools. DeepTox talks about the toxicology. You can also have your own tool. You can just give your own name with some fancy name, link it to your discovery. So these are not the only tools. These are some of the tools I just given as example. AML is like a language, right? Once you have the basics, now once you have the pharma background, now you can have your own tool saying that I'm going to make my tool is going to predict so and so. It can be a clinical application, it can be a discovery application, it can be even a pharma education. If you think that there is a scope, if you want to replace some of the subjects can be taught by the AML, you can have a particular tool built with the existing data and say that this is license. We can also give the license to the people. So what is required is basics of AML. And I feel if you don't learn this, we are the only losers. Ultimately, we are all the losers. Definitely, there is a need for all of us to learn this technique, a tool, whatever you call it, the expertise is going to help you. Whether it's you are going to do the research, tomorrow you want to be in the publication domain, if you want to do the business, everywhere, it is going to help you producing your meaningful data sets. Some of the examples I given here. See, in the drug design level, what we can do in the polypharmacology, in the chemical synthesis, in the drug repurposing, in the drug screening, everywhere, it is having a role. You can just see how exactly it's doing it. I also given the details of it. See how many startups have come in this particular area. Again, see the first one is the blue one, right? This is I'm talking about this USA, right? Everywhere US is leading it now. Why it is leading and why? Again, US only has to have all the startups. And why not India and other countries? See, not even India. One fourth of world population is in India. Not even India is mentioned because it's done by somebody else, right? Asia and where are the Asia and Middle East, right? So this sector, maybe some of the ones. So that means we claim that no, we are IT capital also. We have great engineering colleges. AI is engineering college. Even here also we are lagging. Basically, startups, you don't need a 8,000 crores of money for you to start. You just have to have a tool which is going to build things. I have seen people just with 1 crore they have started less than 1 crore even doing a lot of business in the Gino Valley because now they have realized the importance of the AML in the design level. See some of the examples. Do you know all these companies? None of the company will know it, right? None of the companies will know it. This is one of the pioneering now. Just Excensia, just type it. It will give you how many targets they have been working on them. It's the pioneer in the drug discovery by AML tools. They already have a certain molecules moving to phase 1 and phase 2 trial. And there is this is a in silico medicine. The company name itself in silico medicine. In silico means competitive, right? So they already have a company name itself, and it's I think moved to phase 2 or phase 3 now. So there are already success stories where they have taken the molecule to the next level. So that means it's possible. You don't need to wait for 15 years. You can have a molecule moving faster. We are only talking the AML of last 4-5 years. But already molecules going to fight certain stages, right? So this is one of the example. Where are we in India? We don't, right? Now see the collaborations. Leading pharmaceutical companies and their association with the artificial intelligence organizations. Now you see, Roche, Pfizer, Novartis, Bayer, Sanofi, Takeda, these are all the global leading pharma companies. What are they doing? They're trying to organize this. One of them Excensia, right? These are all the companies. They realize, all the major pharma companies realize that no, individually we are going to be wasting a lot of time and it may not be efficient discovery. Now they realize that they want to collaborate. That's why they have collaboration with these particular AI organizations. So that means there is scope for us to do the things better if you have the AML tools. Right. Let's just imagine you are a pharmacy graduate. You also have AI tools. A Bayer may come to you. Or you imagine design a molecule such that no, I have a very interesting molecule showing a good initial docking scores and limit activity or what are the activity. People will be there to take up your idea. You don't need a lot of money. You don't need hundreds of people. Just need a proper idea. If you can use AML tools and design the molecules, people will take it. You need to have a proper target, properly design the molecule, people will like it. So this slide is mostly to talk about the collaborations, right? This all of since for discovery, protein structures are very important. This is the protein structure, keep on increasing it. One of the important contributors, AlphaFold, right? AlphaFold database. It's keep on increasing, right? Each protein is a target for you. So you may be selecting a new target. So that means there are 21,000, 2,14,000 new proteins are there. Some of them may be a good targets for you. So just have to see which is the protein having a role in the particular disease context that you will be taking, right? See, high throughput screening. What is high throughput screening? We have been talking high throughput. What is high throughput screening? That means you'll have 10,000 molecules. You try to screen on the screening system. It's experiment actually. High throughput screening, I think experiment. That means you need to have the 10,000 molecules synthesized and add to the one of the plates, high throughput screening plates are there. Now then read the results. So it was a very fascinating thing 10 years back, 50 years back. Oh, if you have high throughput, without high throughput screening, your discovery is not possible. But now you don't need to even do the high throughput screening. If you have to do high throughput screening without compounds only. What's your screening now? I don't want to even synthesize it. No synthesize at all. I want to screen at the computer. Every molecule will be put into the funnel. These are my conditions. Molecule having this property out. Molecule having this property desired. Like that you define it. Then filter it. Then give you the set of molecules. Now you don't need 10,000 molecules. You just need 100 to 200 molecules. If you can apply the AML tools. And teach your mission saying that no, these are my desirable properties. I want this particular features to a compound to go to the next level. So you define the criteria, then it will be filtering it out. So now easy for you, right? If you have only 10,000 molecules, only 100 molecules, your chemistry will be so happy to synthesize it. To put more efforts, then you will be able to better screen the compounds in the in vitro environment models and say that okay, these compounds are showing activity, and your success rate will be much higher. That's what is the data is talking about. I have a friend in the Genome Valley. He says that he had done more than 20 projects. All projects are US universities. As I mentioned, I was trying to use one lakh US dollars. Or university is paying 1 lakh US dollars for just the activity, and he says that all the 20 projects, not even one project got failed. What it indicates? 20 projects got successful, right? So I was trying to work with him. So tomorrow I will also have molecule moving like that, right? So it can be structure-based screening, or it can be a ligand-based screening. That's what he's talking about. What do you do exactly here? The structure-based drug design. You will have a protein structure known to you. This is a crystal structure of protein, or what are the 3D structure of the protein. Then you try to virtual. What is virtual screening? What exactly virtual screening is? Virtual meeting, right? So you don't do the actual screening. You just put your protein is there. You try to see the how your different chemical moieties structures are available will be interacting. That's what virtual screening is. And some of them are going and having some interactions. Some of them will never be having interaction, and will be only interested in which are the molecules are having interactions. That's what is called the hit. From the hit, you try to okay, this is the hit. I know that okay, this is the one actually having some interaction. Now we'll try to optimize the lead. The lead will be further chemical synthesized and become a move on, right? That's what is the structure-based discovery you're talking about. This one side is structure-based drug discovery, drug design. Other side, ligand-based drug discovery. What do you do in the ligand-based drug design? Ligand. Ligand means agonist or antagonist, right? So you try to play with the ligand. You try to modify the heterocyclics, or you try to modify the side chains, whatever it is. So you try to do the ligand-based drug design as well as structure-based drug. Both are important, right? And both will be working together to have a new drug discovered. Right? That's how it will be done here. Just see computer aided and computer driven. We have been talking about computer aided. What is computer aided? We have been taking partly, okay, computer support is required here. But now it is a computer driven. Everything is computer driven, right? Earlier, our train tickets and all the flight tickets used to be manually. It's all computer driven. Now, similarly, your all the activities should be computer driven. That means not that everything you replace it, but initially you do the computer based modeling, a computer based simulation. Then you try to prove the same in the animals, in the models. That's how it is. That means computer support followed by the experiments. Remember, you may be having a fantastic computational tools. Until unless you prove experimentally, it's not 100%. It only go close to that. But until unless you prove it, it's not successful, right? You need to experimentally prove it, mainly for the pharmaceuticals. As I mentioned, toxicity is one of the biggest concern in with the drug discovery. We can predict the toxicity much better with the AML tools. In fact, there are a lot of tools. And all the pharma companies, whoever is doing APIs and a new drug discovery, use these tools. That means you have chemical structure, put into the computer, it will tell you that the molecule is going to be safer or not. What kind of toxicity may be having it. All the issues will be trying to solve. It will predict it. So the one which are having a problem, gone. It will not be taken further, right? So ultimately what is required is, we don't want to spend 2 to 6 years of time here, not 5 to 10 years of time here. That means it should be reduced significantly. From 15 years, if you can reduce to 5 years or 3 years, it is going to be a very successful model, and it's very only possible with AML. Every pharma company are now trying to work with AML. Now, for us to go with the current trend, you need to have the basics of it. At least you should be convinced that no, okay, AML is the only way I can be able to succeed tomorrow. Whether you do in the research, whether you do in the something else. And me, pharmacologist, I'm very happy working with animal models. I don't know to talk about the AML, but we realized it after publishing 200 papers from Naipur, Hyderabad, that no, I'm nowhere discovering anything, because it's all just something is giving, I'm doing an activity, I don't know what exactly happening it. Now, if you have an AML tools, where somebody is supporting you, if not me, my collaborator is supporting and making that, okay, this moiety is optimized for all the conditions. It is having a good binding to my target. It's having also safety profiles are much better predicted. I'm more convinced, right? That no, I can have a confidence to take this molecule, do the in vitro studies, say that no, okay, this compound is effective. That exactly, I am trying to do now. So we are trying to use a new targets. Now we have to spend a lot of time to understand which target is good. That's where you have to read a lot of literature. Literature reading is very important. The articles you have to read it. Okay, it's not textbooks. Textbooks are fine, but textbooks won't tell you what textbooks are talking about 10 years back information. But current is AML trend, right? You should know the articles read. How many of you read the articles? Until unless you read the articles, that means you are just following the old trend. You actually, it's good to have a old trend followed. Basics are important, but plus combined with your current trends are important for you, right? Just see hierarchy of the artificial intelligence. I have no idea on this. I just asked one of my students support on this because this girl with a informatics background wanted to do PhD with me. From Hyderabad, she just finished informatics. She just want to work with me. She's waiting in Kakinada. She want to come back to my lab. So I asked her, let's write the review. She wrote this article. Make this figure. See the artificial intelligence, machine learning, and also machine learning have different reinforcement learning, supervised learning, these are all the cores of artificial intelligence. It's data of it. So what exactly is? If we have this particular basics learned through any program, these are going to be really useful to you. Trust me. So you all should get this particular course enrolled somehow. Spend some time. Tomorrow you'll be very, very, efficient in doing the things. Okay. You walk away from this particular field, particular area, you'll be later regretting it. And again, it will be asking you definitely to learn later. After 5 years, learning today itself, right? It's going to help you tomorrow, right? It's a simplified version of it. I said this is very complex. She made in this slide. So artificial intelligence, machine learning, deep learning, so many things are there. And each and a drug discovery, okay, if you want to talk about repurposing, one tool is used. If you want to talk about protein structure, one tool is used. Like that, there are tools are available. And for you, I just gave you a deep rocks and deep chemistry. These are all the tools built through this boxes only, right? This is so far the general talk, right? So let me just talk about the research. Though research is not good for you to do so much, I just talk about my research, what I have been doing it, so that now we'll complete it, right? So drugs can come from all the sources. We have been talking about the AIDS. What is AIDS? Acute respiratory distress syndrome. We all know during COVID. Also called SARS. Severe acute respiratory syndrome, whenever the foreign particle like a virus go into the lung, your lung will be trying to respond to send the immune cells, inflammatory cells, so that the viral will be inactivated. But that exaggerated process of the immune cell infiltration into the lung will be causing a severe lung edema, inflammatory condition. That's why your lung will be highly inflamed. So it's not the COVID which is killing. It will be too much hyperinflammation. So this is the condition we are working on, and there is a excessive inflammation in the lung. For long term, it will be causing a pulmonary fibrosis. You might have studied about it, pyriferinone and nintedanib is the drugs. So what happens in LD lung will be having a alveolus and the capillary only small barriers, whereas here in the pulmonary fibrosis there is extracellular matrix accumulated. Due to this extracellular matrix accumulation, the gas exchange is not proper. That's what is happening in the respiratory failure. So this is a chronic condition of the lung inflammation. So we work in this particular two different areas, where inflammatory phase followed by the fibrotic phase, where your extracellular matrix will be, the blue fibers will be accumulated. That's what at the molecular level what happens, at the cellular level what happens, I'm just mentioning here. So when there is a COVID virus, it goes to the lung, produce a lot of lung acute respiratory syndrome. It also causes pulmonary fibrosis. So post-COVID complication, the pulmonary fibrosis basically, right? It can also cause other organs. That's why we are talking multiple organ failure during COVID was reasons. So all this process is happening through the inflammation. That means inflammation is we all know inflammation, COX-2 and another the prostaglandins, right? We may have different COX and other regulators, and so that is a big villain. So out of this, I'm talking the netosis. What is netosis? Netosis, neutrophil extracellular traps. There are neutrophils. There is something which is coming out of the cell, extracellular traps. This is nothing but some part, neutrophils are multi-lobed cells, multi-nuclear lobe cells, right? Some part of the DNA, the lobe, will be coming out of the cell. That particular part of the DNA is activating the immune system, triggering the inflammation. That's why it's called the netosis. So what I'm saying is, not just COX today, you also have another target in the form of NETs, right? So NETs can be one of the important mediator of the inflammation. This is a process when there is a particle entering into the lung. Neutrophil will be going to the alveolus and trying to send some part of the DNA out of the neutrophil and make a meshwork like of structure that's called NETs. So NETs are the mediators of the inflammation. That's why I'm saying it here. Neutrophil infiltration to the site where reactotoxin species are also produced and also releasing so these all NETs will be having all these components. Basically, these are the defensive. If you have this component, your foreign microwave particle will not be able to survive. Okay, it is doing the function. But what happens? The excessive release is trying to attack your own system. That's called the immune system or inflammatory condition. So this particular netosis process is controlled by the enzyme called the peptidyl arginine deaminase. This is the new target for me now. Next is controlled by the PAD4. PAD4 is a new enzyme for me to inhibit. Now understood? Now I want to inhibit the PAD4 by 10 minutes. So I want to inhibit the PAD4. That means PAD4 is my new target. So we are now, as a pharmacologist, I say that no, the PAD4 is my new enzyme. I wanted to inhibit through my chemical approaches. It is having a role in not only just the lung conditions, sepsis, arthritis, fibrosis, as well as the colitis. In all the disease conditions, NETs having a role. That means NETs can be controlled, or the netosis can be controlled by the PAD4 inhibitors, and the NET degradation by the dornase alpha. Dornase alpha is nothing but it is used in the cystic fibrosis. Just mention Googling the dornase alpha. It's a drug for the cystic fibrosis. There is a reason why you are using dornase alpha, the DNase in this particular condition. It's nothing but the cystic fibrosis will have very thick sputum. Sputum is thick because NETs are there. NETs are DNA. The DNA is very thick, viscous material, right? Now, if you give that DNA, which is going to break the fragments of DNA, it will be thin sputum, so that your respiratory block will be controlled. That's why it's approved drug. So you can have this particular approach. We made a nanoparticles approach as well as the PAD4 inhibitors to prove it. And there is a paper here. Suggests the title: long-acting nanoparticle DNAs for effective substitution of the SARS-CoV-2 mediated neutrophil activities and cytokines. So there is a paper published in the biomaterial during the COVID. Based on that, we have designed a molecule. There is one nanoseria as a one nanoparticle, and the dexamethasone loaded P as a nanoparticle in the system. Then we have decorated the surface with the DNAs. That means not just DNAs. I have another layer which is nanoseria, which is a nanoparticle of very good antioxidant, anti-inflammatory. Dexamethasone, we all know it's an anti-inflammatory drug. So we try to make this nanoparticles. This particular approach we have submitted for the ICMR proposal. We have a fund granted for this particular approach, and this we have been submitting now as a grant. Now we need to have animal model. We have the animal model to do the lung conditions. Then we have the tool to do how the lung is functioning. We have the whole body crystallography for me to understand it. This is the experimental model for me to how I exactly do the my ARDS model and pulmonary fibrosis model. Then we have proven my smart nanoparticles what we have made in the LPS model, lipopolysaccharide inducing model. When I put the LPS into the lung, it produces a lot of inflammation. Now I have given my nanoparticles, smart nanoparticle, I claim it, protected from this condition. This is the inflammatory lung and how they have already talked about. It's a histopathological data also showing that it's actually protective. Another condition is which is also causing a lot of inflammation, the sepsis. There is no treatment for the sepsis basically, and one of the organ getting affected is the lung. When there is a sepsis happens, there is no treatment. Not even your anti-inflammatory drugs are effective. So we have this model in our lab where you just puncture that cecum, so it's going to release the contents of the peritoneal cavity and cause the inflammation. And we see the lung which is having a very severe inflammation, and our nanoparticles have reduced it. So we have proven that through different molecular studies. Finally, we also, since I was claiming that PAD4 inhibitors are overexpressed in the lung conditions, In the pulmonary fibrosis, we have seen that the normal lung and the brown color is increasing the bleomycin into the pulmonary fibrosis. Western blot also showing that. What I'm saying is this particular enzyme is overexpressed. That means it's a good target for me. Now we try to study the standard inhibitor chloramidine, which is protecting against the. So this is the paper we have published it. Based on that, we try to design the new molecules, and we have the in vitro tools, because whenever you talk about the new molecules, you need to have an in vitro tool. So this is the in vitro tool for me. This is a normal cell. When I just stimulate with the PMA, which is causing nets. These are the nets. That means I have the tool. Then I also have a protein data bank for us to understand what kind of binding sites it has. Then we have standard inhibitors proposed by different companies. We understood what kind of docking score they have, and we have designed the molecules. Out of these molecules, one molecule is showing some good inhibitor activity. So this is our new molecule. We have designed it. Then we also done the molecular docking study with the M4 is our identified molecule. We also done the MD simulation studies to see what kind of hydrogen bonds and other interactions are happening it. Then we also did the in vitro flow cytometry based screening of all the 5 compounds which we have identified. Then did the in vitro study saying that no, okay, this is the positive control where we are seeing a lot of netosis, and the compounds including the C14. 36C is a C14 is also decreasing this green fluorescence. That means it's actually protecting from the netosis. We have done the study in the AIDs model, which is a animal model, then followed by histopathology. Then we also did the study in the pulmonary fibrosis, different parameters we have evaluated it through different biochemical assays. If this histopathology, see the lung of the disease animals, HNA staining, normal histopathology, and the MT is nothing but magnesium trichose staining. These are the collagen. That's what I'm saying. Extracellular matrix is highly accumulated in the lung. This is not a good condition, right? Now treatment has resulted in reduction in the this particular condition. Expression of the different proteins, collagen, fibronectin, fat, these all increase in the disease control, and treatment has been reducing it. Now we may have a molecule. Unless you do the toxic studies, the molecule is not safer. That's why for us, you need to perform the toxic studies. Safety is important than the therapeutic activity. We try to do this, and we can predict the safety through the computer-aided drug designs. And even if you want to treat that lung condition through the specifically, you can also inhalation drug delivery. No oral treatment, no IV treatment. Just put the drug into the lung through the inhalation route. It is going to be selectively going to the lung. Just see the example. Alphazer, right? Inhaler for the insulin. We are thinking of insulin diabetic condition to the lung. Why not for the lung condition? That's what is we are thinking of. So we can have a localized delivery based lung exposure and so on can be done. Just imagine, India is having a tuberculosis, a big problem. We have been taking the drug orally only. Why not lung? Why not inhalation route? Why you don't have the product? We can have 3 drugs combined inhalation, right? Why can't we have it? So this is still an opportunity for us, right? I feel there is a lot of scope for us to do it. Now these are some of the advantages and disadvantages I talked about it here. There is this particular characteristic important for you when you are talking the inhalation. MMA, median aeramic diameter. That means which part of the lung which is going will be decided based on the MMAD. So you need to characterize that there is a cascade impactor. How many of you know the cascade impactor? There is a book in the pharmaceutics book. This cascade impactor is there, a tool. So it will tell you which part of the lung your drug is going. So this is an instrument where you just put the inhaler here. It is trying to go to the different stages. Which stage will be telling which part of the lung it is accumulating it. That means your microbacterium tuberculosis will be stage 4. Where the drug is going there or not, you will be able to predict based on this. This is the FDA requirement, compulsory you have to do it, right? So this is the tool we have it in our lab. So at the end, I just want to say that yes, as at NIPER, I may be doing a few things, but as a faculty sitting in some of the different colleges, what kind of activity can be it? For you, what is important? You cannot just take a dream project like cancer and say that, oh, I don't have the tools. You need to take the simple tools. Just take simple psoriasis, allergic contact dermatitis, burns, wounds, arthritis. These are the very simple models to induce it. Anybody wanted to develop these models, come back to me. We can help you. We can even demonstrate you what are the models you want to have it, so that you'll have a good team for you to chemistry team, formulation team, all can work together for better publication and better patent. It's possible to do the discovery or the development from the institutes like Viper or any other institute, not necessarily NIPER it should be, right? So I think with this I will try to thank NIPER as well as all of you for your patient listening. I know I have exceeded the time, but if you have any questions, I would like to take those. Thank you. I'm sorry, sir. You have been hinting me, please stop this topic, but no. See, yeah.
Thank you, sir, for your sharing knowledge with our delegates and provide any questions.
Yeah, what?
Stage is open for you all. Any questions from audience?
Thank you, Nikhil. Yeah.
Thank you for your wonderful talk, especially a big intro about AIML and also how you have been using some of those for your own research. I really impressed and congratulate you on the new compound of PAD4 inhibitor. Right.
Yes,
I'm
yes.
curious, how did you come across this compound? Did you systematically synthesize a set of compounds for screening, or what kind of initial data set you used to do virtual screening?
So for this, what we did was, no, we have used that, and if you see the, we have studied how the existing inhibitors are actually showing the activity.
Exactly.
We studied that first. Then we understood, okay, these are the set of chemical moieties required. Then we did the computational studies. Basically, we have done the virtual screening by our mixture chemistry people. Then they have identified some molecules. That means we have docked the studies, done the molecules initially, then only we synthesized it. It's not that we have synthesized and did it.
Exactly.
What exactly people are doing is they synthesize the molecule and go away docking. No, we just did the docking, understood the molecule having a good score, then we synthesize them.
You design the molecules in silico first, then you find the best ones and do the synthesis for the best ones.
Yeah, and you don't need to congratulate me. This is just initial discovery. We have not to do that AML tools. The missing link here we are not in the AM because we don't have expertise. Now I'm just giving this data to one of my collaborators. Can you further optimize it?
Definitely. I think you can further use AIML tools to optimize the ADMET parameters. And this, you are the lead, right? And you can go to optimize it.
True, true.
Anyways, I think there's great hope from your results. Yeah.
Thank you.
Thank you.
And I know a lot of inflammatory model conditions can be cured through this. We have been depending on the CAPS too, right?
Multiple diseases.
So that's what.
Same target.
Exactly, yeah.
Very nice. Thank you.
Thank you. Thank you, Nikhil.
Any questions?
Students, you should ask questions. At least you should come to NIPER. How many of you writing GPAT? Come on, you all should write GPAT and come to NIPER. I know. Why is you all applied or already qualified? Writing now. Good, good. Prepare well for the exam because without GPAT you will not able to write the NIPER exam. Okay. Why I'm saying NIPER is, you will get a fellowship. The culture is different there. You have a lot of time. You can do a lot of, professional developments. Okay. So the infrastructure is very good there. You all should, it's in Hyderabad, right? Many people are from Maharashtra. 70% of the people are from Maharashtra. Unfortunately, I know at least I can remember now. Many people are from, not many Telugu people basically at least. What happened, I don't know. But when I was in Mohali, I used to have 100 people from Telugu state used to study in Mohali. Out of 200, 100 people used to be from Telugu states. Today when I see in Hyderabad, I have seen not many people. What happened? Motivation is gone. Pharmacy has not a feature. It may be, but you're already in the field, right? You should go to the next level. So you should come to NIPER basically. What I'm saying is write GPAT and try to come to NIPER. Okay. When some of your faculty, Vidya, you see, both have done in Kolkata, right? It is Hyderabad, right? You need to go to Kolkata. Why you should not try? I went to Mohali to do with no Hindi. The people used to make a lot of fun of my Hindi. So yeah, so survive. And today I have a best of the best friends in Mohali compared to anybody else because your difficulties will make you should make into opportunities. So I feel that, right? So you should don't scared of this. You may think, okay, artificial intelligence is not at all my business. No, it's your business only. Everybody is talking about why we should not. I try to demonstrate, right? We can do things through AML. For you to get a good job in Hyderabad and Novartis and any other company, you need AML tools. Just write a AML skill in the resume. Of course, they'll ask questions on that. You'll be picked up. There are not many people having this background in the pharmacy. If you have command, you will have a lot of the job opportunities. Okay, so you should insist, sir. We have engineering course, right? We can just enroll in the course, audit course, whatever course, you just attend the course, right? Yeah. No question, Namal sir. These are B Farm candidates. There are some M Farms as well. How many M Pharmacy people are there? No M Pharmacy people at all. Exams are going on. Okay.
Yeah. Good morning, sir. Do you have any model for melanin content, sir? Like we developed a good formulation, like
Okay.
it is reducing the patches on the skin.
Vitiligo you're talking about, right?
Yeah, not vitiligo, sir.
Melanin
Like
vitiligo.
only we can say like
It's a vitiligo. It's a condition is vitiligo. It's a condition.
vitiligo is the white, sir. That is the different
Okay.
vitiligo.
Now you're talking about.
I'm talking about the melanin content.
Okay. Okay.
Yeah.
We have
Yeah.
essay. We have
Like the
essay.
inspectors, police, we can see they exposed to the sun for a long period of time. They develop some patches, black patches.
Correct. Yeah.
So that is reducing very well with our formulation, but we would like to go for that
You
model.
want to estimate it, right? Melanin content.
Yeah, we would like to prove that in animal study or any model so that before going to the market.
Now we have one model. This is vitiligo model, where you say nitrobenzene and some compound you apply to the skin for 1 month to
Okay.
reduce the discoloration of the skin.
Okay.
Now we are trying to treat that condition.
Mm-hmm.
So that model we have it, but we may have to see what exactly the model is.
Sure,
If the
sir.
model is there, we can reproduce it.
Okay.
We just have to apply it topically, right?
Yes, sir.
The cream, whatever it is. We
Yes.
have that model, but for the induce the discoloration we have.
Okay, sir.
So then we try to see how the compound is protecting. I may have to go and exactly I will not have all the models. But the literature is available, you can do it.
Okay, thanks.
Yeah. Good. All the best. No, the girl is trying to go somewhere. So we will have a inhibitions not to ask the questions, but you should willing to ask the questions, Let it be a simple question, right? No need to be a very scientifically very strong question, right? Every question is important. But how many of you wanted to really become entrepreneurs? Or you have one, okay, your faculty want to do it, but what about you people? It's the era of the startups, era of the entrepreneurs. India is really trying to boost this particular, field. Every budget is, strongly proposing to have a startup culture in the country, and pharmacy should be one of the area which you all can jump into. I was just attending last month BioAsia, right? But you know the BioAsia, right? All the startups are with MSc background. I was surprised. MSc people are talking of the drug discovery, vaccine. We people, pharmacy, not many people are there with the pharmacy background. What are we doing? You know everything about the drug discovery. You know what are the side effects, what are the receptors. You know everything, but we are not dreaming. We are actually, imagining our thing very less. Our people less, and you have a lot of capabilities. I feel kind of subjects you study much better than any other subjects, right? You all should think big, dream big. Okay, mainly the girls. It's not just to do the studies and done. Government is trying to support a lot of women entrepreneurs, right? You take the money at least from the government. Since you are taking the money, you will try to do something, right? You never know. You start something, you may get something else, right? Okay, enough. Thank you.
Thank you, sir, for sharing knowledge with our delegates and providing a glance of drug discovery and development.
I request Principal of Piper, Dr. A. Ramesh, to felicitate our guest, Dr. Chandra Godgu, with a memento.
Thanks.
Right. I think thank you so much for your wonderful patience in listening for more than an hour. I know. despite all of your heart is partly there, right? But one important thing which I want to make is, I think I don't know whether you have followed what Bill Gates has told just one day back. Are you listening to the news around? So we biologists are opting and looking for AI. And you know what Bill Gates told? The future AI is going to change, but it is going to replace all the IT jobs. But there is one area where AI cannot replace is the biology. So scientists are the ones who cannot be replaced. So in future, all the MPC taking students, right, they will be now opting for biology because biology is a place. So future is not only technical, or you, what do you call it, as electronic digital, you tell, right? But I want to share that future is only biological, not, no more technical or digital future ఉండదని clear గా Bill Gates has told. So because AI cannot judge or AI cannot do experiments, it can only take the data, interpret it and outcome. It can accelerate the process, right? It can help the project, but we scientists have to do the experiments. So it's a bridge between technology and scientists which can ensure the better result. So thank you so much, Chandra. Although I have stopped you, sorry for that, right? But because these are B.Pharm students and more of depth of technical will make them bounce. So
At least I could see, they're not at least watching the mobiles.
yeah, that's a good part of our students. We always have that sincerity in all of them, and happy to all have all of you here. Thank you so much once again. Thank you.
We are happy to say that a good number of sponsors played a key role in the success of this event with their contribution. I take the opportunity to thank them by appreciating with a memento.
I would like to request M. Narayana Raju from I-20 Fever. Present.
Now I would like to call upon P. Govardhan Reddy, BSc Books Private Limited. Now I would like to call upon Mr. Shah, Managing Director, iCode Healthcare Solutions, KPHB, Hyderabad. Now I would like to call iCode Healthcare Solutions for a presentation. Request students to be please seated.
Good morning, everyone. I won't take much time. I think we are running out of time and you're rushing for events. So I will like to consume more or less 6 or 7 minutes. I'm promising that, and I thought I'm that. Okay. So if you are someone who interested in PhD research, this is not going to helpful for you. I'm repeating again. If you are into PhD or explore the science in a scientific background, like PhD, doctorate, fellowships, this is not for you. If you are someone who want to pursue your career in informatics, as now the respected principle of your Viper said, biology can't be replaced by air. If it is biology is combined with AI, the deadly combination, right? Yes or no? So, if you are someone who feel very jealous by seeing your brother or sister or your nephew or someone, anyone, because they are doing great job. Okay, so I will manage. By seeing like work from home kind of opportunities, like cafeteria, very set technical things like they are going by flight, choosing flight instead of bus because they are paying well, getting paid well, right? Remote area workout locations, work from home, complete day shifts. If you feel very jealous by seeing that, you don't need to feel jealous from this moment now because if your aim is to explore the world by earning by yourself, if any of the above criteria you have ticked the box, then medical coding is for you. Exactly, this profession suits for you very well. The deadly combination of beauty of this is you can explore the world by choosing science, by combining with information. So the information is the great tool, right? But so in this program, we'll talk all about the medical coding. So how many of you are aware of about medical coding? Just raise the hand. So from that I can. Okay, very few, very less. That's so bad. So then I won't start with ancestry or history. So medical coding is a type. How many of you are aware of insurances claiming procedure? Okay, very less. That's fine. Have you ever felt or have you ever heard that I will or the software or anything will rule the world? Rule the world and rule the world. Both. Have you ever experienced or have you ever heard? Oh, very few, okay. How many of you? Very less. So the same way, both vice versa. Why medical coding is the best career opportunity? Why? Why we should not choose pharmacy? If you are prone to chemicals, if you want a high-paying jobs, or you're not someone who fit into pharmaceutical industries, then medical coding is a very good opportunity for you guys. And moreover, the best thing about medical coding is work from home sort of opportunities, remote areas, locations, and more. And the incentives, great opportunities. And if you are someone who really want to pursue your career in abroad, but you can't afford the fees or you can't, then medical coding is again the best opportunities. Why? Because you have a lot of exploration and identification in medical coding sector area. Mainly in US, UK, Dubai, overseas. And moreover, but what is the medical coding? And how it is related to insurances? And nowadays, the first question the doctor asked when you visit a hospital, he doesn't ask like what's happening, what you're suffering with. The first thing the management asks, do you have any medical insurance? Is it yes or no? Is it private or public? Are you a government employee? Nonsense sort of questions, right? But that's true. That's exactly how it's happening over there. So the awareness on medical insurance is booming. So that's why I can assuredly say the medical coding is going to be boom in our India too. The India doesn't have much awareness about the medical coding, but from past decade, the medical coding is booming in our India. Why? Because the people are very having lots of knowledge on insurances and all. So I can definitely say if you are not really into pharmacy or research oriented, the health with information with IT sector, which deadly combination is where you can pursue all your dreams. The medical coding is the the best opportunity. And why you should join in iCode? If you raise a question, why you should join in iCode? Why MFL or any restaurant or any paradise stand in business till now? Because of its quality. Exactly that kind of the best ever quality we provide in our education. That's where we stand. If you see the lots of 100% pass percentage and placement we provide, and moreover, but what is medical coding? Again, this I am asking, but I would like to. So what he said, medical coding is the process of converting a patient healthcare information into universal alphanumeric codes. This information can include medical procedure, diagnosis, necessary medical equipment, and medical services. Information from the physician notes. Medical coding standardizes the language and presentation of all these elements so they can be more easily understand, track, modified. Modified. I know, didn't understand anything. So in easy terms, instead of paying bills to the management team, we just claim our insurance through these codes in simple terms. So this is all happen in medical coding. So is it tough? We are like paying, like they are paying good amount of salaries and providing remote working areas, flexible timings, including incentives. Is it tough? Not really. Not really. Exactly not really. The flexibility of time is also the flexibility of working procedure. Like I doesn't say like just give it a code. But if you are have enough knowledge, then it will be the cakewalk for you all guys. It will be like the smoothest and soothiest way you can work and rebuild your career. In our institute, we provide 3 months of course in this daily one. The flexibility of timings also we provide based on like evening batches, morning batches, offline course, on online course. Through the 3 months, we first 2 months we provide the 1 to 1.5 hour class session where you explore the lot of medical coding procedures and codes. Over there, you get a lot of knowledge over there, like how you have to code for each and every diseases. And after 2 months of completion of the 2 months, again we will give you a practice session for 1 month, and then you will sit for the exam. In this complete 3 months duration, you learn about the ICD, CPT, and HCPC systems. And those, the beauty of this CPC exam is open book exam. Even there is no intellectual student, there is no lower backbench student. Everyone is intellectual over here. If you are able to write the English and if you are able to understand it, then it's exactly you are going to grant the CPC with our guidance. It's a promise from our side. So only the thing is, just you have to know what he is asking and what I have to do. What he is asking and what I have to append. That's it. Yeah, as now I've discussed, flexible schedules, remote work opportunities, career opportunities, highly rewarding and get paid. I hope, do you have any queries or questions about this? Like anyone from student sides? I think I have more 2 minutes. Yes. No questions? Actually, we even have a slot over there. If you have any doubts, we always open and we'll give the pamphlets to you. We'll share our numbers. So if you're really into explore the world with having good paying jobs, then exactly medical coding is for you. And our institute always have a open and warm hands for you. So you can contact us. And if you want to ask any doubts, but you couldn't ask over here, we have a slot over there. You can come and ask, know the details about our institute personally, privately, and if you're really interested, then come, join, explore the world, rebuild your career with our institute. Thank you. Thank you for the opportunity.
Thank you, ma'am, for your valuable speech. I request everyone to please enjoy the stalls and have lunch, and we'll meet at sharp 2 p.m. for valedictory session. Thank you.